So we're trying to do something that's not been done in recent times so much, which is to get together people thinking about theoretical ideas, mathematical theoretical ideas in aging. So I'll use the whiteboard. By the way, Max, it's a 20-minute thing, right?
I have a question. No?
Yeah.
Can we get the stream?
The stream over there, so you guys can see the whiteboard. Yeah. Yeah, so I think what is physics good at? One is connecting the dots between many different facts. Second is physicists are trained or cursed or blessed, I don't know, with the belief that there's simplicity inside complexity, that there's some angle where things look simple. And that leads you to try to make assumptions that are as simple as possible. And then once you make those assumptions, the math proves the conclusions are correct. The assumptions might be wrong. Then you get quantitative predictions. And a good theory is one that makes you do new experiments. And usually that helps improve the theory, and you get a good cycle like that. So it's the seeking simplicity, connecting the dots between lots of data, and having a way to generate a lot of quantitative predictions. You keep trying to falsify the theory. So I'm going to give an example here, right? So how to build a model of biological damage production and removal, death and disease, that aims to account for as many of the known quantitative patterns that we have in aging. And it also has universality. So it'll have X, which is damage, but it's agnostic to what X is. And so you can connect fruit flies to worms. Each one will have a different kind of damage. And then it also gives you ways to figure out what the chemical nature of the damage is by the properties of the survival curves or things that you can measure. Right? So at the end, I'd like you to know this model, the saturating removal model, and what it can predict and what new predictions it makes. Of course, because the assumptions are so simple, it's just fiction, but maybe hopefully useful fiction in making new experiments come true. So nice that you're here in here. So what are the quantitative basic facts of aging? That we want to explain. You can say, or uses constraints to build a model. So this is age T. This is survival. How many organisms survive? Let's think about what's not aging. You have an island with some 100 organisms, and they don't age. They're just eaten by some predator at some constant rate. So what will be the survival? Like radioactive particles, survival will be exponential, the number of organisms remaining. But if you look at what happens in human beings, survival in other animals, survival curve doesn't look like that. Young organisms don't generally die, and older organisms do. That's aging. You're different at age 20 and at age 70. Right? So there's the median lifespan. It's also very useful to talk about the risk of death as a function of age. The risk of death, by the way, is how many organisms die in a certain year, divided by the number of surviving organisms. And that, log H, there's lots of death when you're young. There's a minimum around age 15. Then there's a plateau. This is deaths of stupidity when you're teenage. And then there's an exponential rise and another plateau. Or actually, when we look very carefully, it's actually rising here, but that's a subtle point. If you don't discount the depths of stupidity, it actually looks exponential all the way down. So several decades of exponential. That's called the Gompertz law, where the risk of death goes up exponentially with age for a few decades, and then it becomes kind of around age 90, something like that, starts going down, and above age 100, it's about 50% chance of dying per year. So what do you say to someone who's 120 on their birthday? Have a nice day. Joke. All right. So look, we want to explain all these patterns, right? Now, what happens when you take fruit flies and you grow them? You get the same thing. Right? In mice, it looks slightly more curved, and in worms, it looks more curved, more like a Weibull function. What happens in midlife if you take fruit flies and move them? No, if you grow fruit flies on a better diet, they die less, they live longer. What happens at midlife if you shift them to a better diet? Sins of the past are forgotten. Yeah. If you shift into a different temperature, by the way, it's only the slope. So what's going on here? We want to explain that. So there's a slowly expanding, some accumulation over decades that gives you this H. And then there's a fast time scale where you can shift down. Also, people who move, when West and East Germany were united, it was like these fruit fly death curves. And within a couple of years, the East Germans went to the West German better survival curve. Isn't that interesting? So there's a slow and fast time scale. What else do we know? Death is stochastic. How do we know that? First of all, organisms die at different times. This is not a step function. Organisms die at different times. Even genetically identical organisms, like grown in the same conditions, like genetically identical worms, flies, mice, they die at different times, almost like two unrelated individuals. Also, identical twins die at different times, almost like two unrelated individuals. The heritability of mortality is, does anybody know what the heritability of the time of death is? 20%. So it's not genetics and not environment that accounts for 80%. What is it? That's another thing physics is good at, stochastic processes, all the history of Brownian motion, Langevin equations. So we'll use that, stochastic. Okay, so all these facts we want to figure out. Nick Strostrup, who's going to speak here, discovered something interesting. When you take worms and give them, let's say, a better diet or a lower temperature or some genetic alteration like DAF2, one gene can make them live longer, right? That's the longevity field. You look for survival where you give the intervention and you live longer. You shift the median lifespan. What he discovered is what physicists love, data collapse. If you divide time by the median lifespan, so now 50% survival is at 1, these two curves collapse on the same curve. That paper 2016 is what got me and also Omar Corrine, who's here, whose work I'll describe here, when he was a graduate student, into the field. Because before that I thought aging was just, entropy, decrepit, lots of processes, messy, not for me. Once I saw that, I said, look, there's an elegance here. Maybe these interventions, like a DAF2 mutation, is changing at one time scale. And it's like scaling aging. So if you look at an old mouse, old human, that dies in 2 years. This is in 80 years, but you can tell it's an old mouse. They have a lot of the same declines, very similar, right? White hair, kyphosis, cataracts. Am I right? So scaling like that, how do you understand this universality or near universality? Of course, some organisms have different curves, etc. Trees live longer. So scaling we understand. So all this is enough quantitative patterns for physicists to constrain a model if it's simple enough. If it has 100 parameters, you can't. If it's simple enough, and that's very exciting that was very exciting for Omer Karin, who's here. His paper. All right. In order to make progress, though, it's not enough to have demographics about diseases and death. You also need to measure the damage, because in biology, now we're connected to biology, we know it's accumulating damage. There's kinds of damage that are not removable. There's kind of damage that are removable. And there's so many theories of damage. There's like 400 different theories of aging, it was counted at one time. How do you make sense? Which are important, which are not? So you need to measure damage. So the model I'll tell you is based on measurement of damage in 3 different organisms. They come to the same model. Right? So here we're going to plot damage as a function of age. So the first kind of damage is mice senescent cells, which are dead cells that are damaged and they secrete inflammatory factors. And these mice produce photons under the P16 promoter, which is kind of a proxy for how many senescent cells they have, measured longitudinally. So you have these curves like this. When damage exceeds a threshold, the mouse is dead. Another is bacteria. This Yifan Yang, who's going to speak today also, measured how bacteria age and has a dye that goes into the membrane. It becomes fluorescent when you etch the DNA. So you look at membrane loss of membrane potential, you get damage that goes up, reach a threshold, and you're dead. And the third kind of data is frailty, human frailty data. When you take a list of deficits and you ask how many of them a person has, that's also a curve that looks like that. You reach 0.6, 60% of deficits are not compatible with life. It also has these same statistical properties. Yeah. Okay? So these 3 organisms, 3 kinds of damage or proxies to damage lead to the same model. And the way you do that in physics is you try a class of models. So I'm going to write here, rate of change of damage is production minus removal plus noise, right? The 3 things we know we have to have. And you try different mathematical terms for these until you get a good description of these statistics. We know how to do that. And you end up with the following model. I will write the equations and then I'll give you a metaphor that everyone will understand. Production rises linearly with age. This is a kind of irremovable damage that's accumulating. So when you're 20 and when you're 70, you have more of that. Okay? And then there's removal. But we know from biology that biological processes saturate. If you have some removal mechanisms, once you have enough damage, it's going to saturate. Why do we have enough damage to saturate? Because the production rises with age. It's going to saturate any removal mechanism if the cell doesn't know how to make more of it, or the organism. So you have a maximum removal rate, a production rate, bam, you have a crisis. If there was no noise, everyone would die at that crisis point. But there is noise. So noise can make you cross that threshold earlier or later. And then we need to add, because to wrap up our ignorance about the noise, we use the simplest thing, Langevin noise, like Brownian random walk. That's it. This is the model. And you die above a death threshold. So it's a first passage time question. Who dies here? Who dies here? Who dies here? You do the math. The risk of death rises exponentially with age and saturates at old ages, just like that. You do the math. You get these survival curves. If you change something, let's say you increase removal rate or reduce production rate, you get this rapid change because the removal is very fast. Okay, let me give you the metaphor. What's going on here? There's a village which has houses. These houses produce garbage. And their village has 100 garbage trucks. Yeah, they remove the garbage, everything's fine, you have some garbage, remove. But this village is growing. Every year there's a new house. That's at ARDD. But it's a strange country. Maybe it's a communist country. In their 5-year plan, just 100 trucks. Soon enough, it's a big city. There's so much garbage getting made that the trucks are saturated. They're so slow, garbage builds up in the street. And in the end, it reaches a level incompatible with life. Right? So when you change, let's say, do some caloric restriction to the flies, the houses reduce, recycle, and the trucks clear the garbage, and that's why you get a quick improvement. Why do you have the Gompertz law, e to the alpha t? It's because you have your threshold and you have linearly increasing production, so the gap goes down with time linearly, but the chance to tunnel, that's to say to have noise flipping a coin in the right direction, exponentially, so the smaller the gap, the rate of crossing is exponential, just like e to the delta g, like you have in biochemistry, tunneling. That's why you have the Gompertz law. If it was t squared here, you would have e to the t squared, which doesn't agree with experiments. So you see how data constrains the mathematical form? Okay. This model has a really weird statistical prediction that turns out to be correct. If you look at the mean damage over time, it goes like this and diverges at a critical time. If you look at the standard deviation, it also goes up like that. But if you look at the relative, the CV, the standard deviation divided by the mean, it actually goes down with time. What does that mean? Old people become more similar in relative terms in this damage. That's different from almost everything you measure. If you measure blood glucose, it goes up, standard deviation goes up, people become more dissimilar. If you measure kidney, creatinine, more dissimilar in relative. We think this X is upstream of everything, and it's the thing that becomes more similar. And then, why is glucose more dissimilar? Because that's genetics and environment. If you have the genes to get diabetes, or the environment where you're eating too much, then that X is going to give you diabetes. Not everybody gets it. That X is going to give you kidney disease, not you, Vera. So that's why we think that reducing CV is a marker of an upstream factor that's driving downstream things. And by the way, human frailty has this, with Sarah Hag, we looked at some biological clocks and stuff. A lot of them have this property, unlike almost any other biomarker that you measure. So that's interesting. All right, so we were very pleased at this point that this model, so simple, can explain all these facts. Also scaling. If you change the parameters, you can, let's say, decrease the rate of building houses you live longer. And it turns out that if you change this parameter, the rate of building new houses, you get scaling, just like Nick discovered. But if you change any of the other parameters, like the more trucks, you live longer, but steeper. So we have a way, from a measurable function, the survival curve, that's the most commonly measured function probably in organism research, right? Yeah. To tell whether our perturbation is affecting production or removal. Assuming that the model is correct, of course, it's a big assumption because it's so simple. Is it one thing driving aging? Is it crazy? Is it very crazy? It could be, of course, that there's a hierarchy, and in fact, that's what we see, of damages. And they're, unlike when you're young, where evolution tunes everything to be equally important, when you're old, in the shadow of evolution, you can have a dominant factor and a subdominant factor. I'll tell you what we think those are in mice in a second. But we have a way to, let's say, look at a mouse perturbation and say, if it scales, it's changing the production rate. If it steepens, it's changing the removal rate. If we know the removal rate, we know the trucks. If we know the trucks are very specific, we know what the chemical nature of X is. Did I explain myself? So you can do a genetic screen or a drug screen for the removal or the production. That's where the theory gives you like unexpected predictions. And then you can look at what happens. And when you look at that data, you find a very detailed story about what X could be. And I'm going to just say it quickly, combining what we did in yeast, which is look at 4,000 yeast deletions and look at their survival functions. And from data by, I guess, Brian Kennedy? So that was amazing data. Thanks for sharing it. And then you can just, so you guys looked at the median. You have all the shapes. Yeah. And when you look at when you do that, you can get the following story, right? So there's actually two ways of yeast to die. The first way is proteinaceous, and the next way is nucleolar. And so the two paths, I'll just say it. You have histoneacetylation that opens up normally untranscribed regions of the DNA, like subtelomeric regions. They get transcribed, but they're not meant to, so that causes R loops and G4, these kind of problems. And that causes double-stranded breaks and death. That's one pathway. That's the genes. For example, Werner helicase and Bloom and all these progeria genes have many roles, but also removing R loops, so that these are like why these genes make accelerated aging in humans. And the other one has to do with translation. You have mistranslated proteins that form prion aggregates, and you have a lot of machinery to get rid of that, like autophagy, but there's one piece of machinery that's esoteric, but happens to be the first garbage can to get saturated, and that's in the mitochondria, an esoteric removal mechanism. But those proteins come up as trucks, and so when you have more and more kinds of prions, eventually the mitochondria one gets saturated. Mitochondria gets disrupted, and then all hell breaks loose. In yeast, there's two ways of dying: elongated buds with mitochondrial problem, or round buds with nuclear problem. And we think that these are the two first layers in the onion, the first X1 and X2. And each one behaves according to this equation, as far as we can tell. And we think now if we go to, so by the way, all the hallmarks are aging here of single cells. This is, chromatin, genomic instability, DNA damage, proteostasis, mitochondria. If you go to mice, we have to add inflammation, hormones, everything that mice have, that single cells don't. So what we think happens in mice is that you have, what are these houses? They're stem cells. Maybe something like this happens in them, right? They make differentiated cells, which are damaged. They can be repaired, but if they're not repaired, they become senescent cells or otherwise inflammatory cells. So we think this X in mice is this accumulation of inflammatory cells. These are stem cells that produce them. And what are the trucks? They're the immune system that has to remove those cells, NK cells and macrophages, and you have just a fixed amount of them. They don't increase much with age. So these senescent cells, which are great when you're injured, they call up the immune system, they stop cell replication until the immune system can clear them. They're wonderful when you're injured. When you're old, you have mock injuries, these damaged cells, damage-producing units all over your body, making tons of localized senescent cell patches, make systemic inflammation, saturate the trucks, the immune system starts to shift with the myeloid shift towards there. It can't do it. And now you have systemic inflammation and all the bad things that these damaged cells do. Senescent cells are very complicated and heterogeneous, and it's a continuum and stuff like that, but let's just mark it like that. So this model gives you testable theories for what might be specific molecular drivers that are upstream of things like diabetes, cancer, etc. Because we know for cancer you need inflammation, etc. Just say that the same model, if you put in threshold for diseases, can explain why the incidence of age-related diseases universally goes up exponentially and then drops at old age. Hundreds of age-related diseases. And it's, again, a first passage time process. And why does it go down at old age? Because not everybody gets the disease. And by a certain age, everybody who could have gotten it already gets it, and you have this decline. So also this data, the same model, so that's a lot of things that one to put on the shoulders of a small equation. And I think it's a good time to finish and just say that in the afternoon, if you're thinking, I can use the whiteboard to do something like this, then start thinking about it, okay? Not like this, but whatever is your thing. Thank you.
Thank you, Uri. We've got time for a few questions, but please raise your hand.
I was wondering about how negative selection fits in here. I was wondering how negative selection fits in your model, because, for example, Hydra is a bag of stem cells, essentially, and there must be a lot of negative selection going on there. That's probably why they are sort of immortal, not?
Okay, so you're asking about immortal organisms?
Well, not specifically only immortal. I think negative selection can happen also in mammals, but to a much lesser extent.
Negative selection with stem cells, what do you mean negative selection? Stem cells die?
I mean that when a cell is heavily damaged, it will simply die. So
So this explanation requires that these damaged stem cells are, let's say, epigenetically modified, but they don't feel it. But the transient amplifying cells feel it. And Hydra, you can also get the Hydra thing if you understand that organisms that grow can dilute the garbage and therefore reach not at a T, but a constant, and then the chance of crossing the threshold is constant. And that's why exponentially dividing bacteria, exponentially growing organisms, trees, can reach actually a constant mortality and not a Gompert. So you can get that also. It's very interesting, yeah. Neil. Neil, I can hear you, and I'll say it back.
First of all, thank you for the blackboard. Blackboard back is really refreshing.
Thank you.
We just cannot show blots and stuff like that there.
I can show blots.
Right. You can.
Here I use Photoshop. Okay.
You can Photoshop.
Yeah. I was around in the 2000 doing blots. I know. I remember the Photoshop.
I just, my question is simple. So we've done a lot with the production and the removal.
Yeah.
And still we have an increase, if we talk about mice, 20%, maximal 40% in lifespan.
Right.
So what is the physics beyond fixing those things?
Okay.
Second equation of thermodynamics, what's beyond it?
You're saying that a lot of efforts in mice have not produced longer lifespans, more than 20, 40%, something like that, right? Yeah. And in worms, it could be threefold.
Well, but a few days.
Yeah, a few days, right, so who cares? In human beings, we have this phenomenon that the survival curves you don't seem to get humans above 120, no matter what. So that's something we're really interested in investigating, and we think that there's in fact, this model would suggest that there's an increase here, not exponential, but linear and then quadratic, which means that it's exceedingly difficult to push beyond a critical age that's eta over beta. In humans, it's 110. In mice, it's like 1 year. So in humans, it's much more than the expectancy. And in mice, it's less. So mice are a different regime. Mice are in a regime where the random walk is very dominant at old age. And humans are in a regime where the production and removal are more dominant at old age. And that makes it a different game, according to this equation, that we have to think of. It doesn't mean you can't do things, but you'll have to work a lot on noise and threshold Those are things that are going to be important in human beings. And what that is, we're ignorant. So what is the stochasticity? So my best guess is because the immune system is the trucks, the noise is when the trucks go on strike. What does that mean? Immune system more active at night to repair. So if you have sleep, stochasticity. If you have stress, stochasticity, like low social economic status. If you have infection, these things are fast time scale events that I think would play a lot in noise in human beings. And to understand that biology is something we're struggling with. And also X-critical, how resilient you are to dying. Medicine can affect that, so it can keep you alive but sick for a long time. So that's how medicine can change survival curves for old people. But how to do that intervention-wise is another challenge. So I think mice and humans, this signals might be in a slightly different regime. Of course, this is just fiction, but it's just a way to think about it. This is, by the way, in physics called the Prandtl number. For those of you, how many of you here studied physics? Be proud. Wow, 10, 20, so like this dimensional. Okay.
One more question.
Yeah.
Diego.
Thank you. So I love your model, and I love that you're trying to build something that is actually falsifiable.
Falsifiable, for sure.
I would like to know if there is some kind of inequality built in, or maybe it's not even needed to be parsimonious, which is almost by evolution, the removal